Right. Okay. I think then I would like to introduce our next speaker, Dr. Brian Meyer. He is the chairman of the Department of Genetics in the Research Center at King Faisal Specialist Hospital and Research Center. He undertook his undergraduate and postgraduate studies at the University of Western Australia. And he has worked at the center in Riyadh for the past 18 years, primarily studying the genetic basis of recessive Mendelian disease. The title of Brian's talk is Population Genomics to Precision Medicine. The floor is yours, Brian.
Thank you so much, Dr. Bayoumi, for the introduction. I'd like to also sort of thank the organizers for giving me the opportunity to present to the group today. You know, we've been very fortunate, I guess, to be in an exponential phase of research and discovery and application of genomic technologies in clinical medicine. But it's useful to sort of go back and to at least take one slide to think about where all this sort of came from, and really. There was a 30-year period that gave rise to the last 20 years of accelerated discovery. I think right from the beginnings in the 70s with recombinant DNA technology and the techniques to sequence DNA, the Maxim Gilbert and the Sanger sequencing, were the spark that really sort of brings us to where we are today. And then of course, as we progressed into the 80s, we had PCR discovered by Kerry Mullis, the chromosomal mapping of the first disease-associated gene by Jim Gazella, and automated DNA sequencing provided by Leroy Hood. The 1990s saw the launch of the Human Genome Project as a consequence of some of these earlier discoveries and efforts. And during the 90s, the Human Genome Project was able to build the genetic and physical maps that allowed the first draft of the Human Genome Program in the year 2001. So I think the journey over the last 20 years should really acknowledge a lot of the hard work that was done in the prior 30 years to facilitate this. So with following the release of the first draft of the human genome sequence, attention shifted from identifying genes to understanding their function in health and disease. And that essentially was the initiation of looking at human genetic variation. The first large project to look at genetic variation was the International HapMap project, which was launched in 2002 after publication of the draft sequence. And you know, I recall during that time actually being offered by David Altshuler for 24 Arab samples to be included within the HapMap project. But for a variety of reasons, that was not allowed at the time, and I think reflects still the underrepresentation of the Arab population in the genomic databases throughout the world. Nevertheless, I think we benefited from the early studies of variation in other populations and with annotation of the genome, and this allowed the systematic mapping and positional cloning of disease genes that were segregating in families. And I think whilst we went from a very small number of genes to now more than 5,000 genes that are present associated with Mendelian diseases, it's also a good time, I think, to acknowledge the effort of the Arab world, and in particular, my friend and colleague, Dr. Faisal Al-Correa, who is a moderator of this session, that has done so much in terms of cataloging Mendelian genes. The studies of variation also gave rise to genome-wide association studies and facilitated large-scale projects such as the Cancer Genome Atlas. The presence of next-generation sequencing, of course, has then launched us into the accelerated phase that we have seen now through things like the 1,000 genome projects and large-scale genome projects from around the world. We heard a good example through the Qatar Genome Project, but of course, when one looks at the UK Genome Project or the Million Veterans Program. It is a small contribution, but a very significant contribution. So in 2015, with the efforts of the 1,000 Genome Project, etc., the Precision Medicine Initiative was launched by President Obama and scientists from the NIH, with the view of wanting to understand an individual's genetics or the variants in an individual, their environment and lifestyle, and to determine the best approach to prevent and treat disease. According to the Precision Medicine Initiative, precision medicine was defined as an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. So our clinical protocols were and are commonly not fully tailored to individuals. We do see examples of individualized medicine now, but largely medicine has been dealt with in binning individuals into groups that were then treated with particular protocols. And essentially prohibitive sequencing costs, the challenges of data collection, the limitations of electronic health records was something that was part of medicine in the past. The environment for delivery of precision medicine has changed. There now is affordable high throughput sequencing and genotyping. There are mobile health apps that allow you to collect enormous amounts of data. We've heard about BIOBANKS and the importance of collecting phenotypic information as well as genotypic information. And now we are fortunate that many of our medical institutions have extensive use of electronic health record systems. So there was a drive for a large number of national genome programs. And I think when we look at this, it's good to see Saudi Arabia and Qatar represented from around about the year 2015, which really, I think, shows that the Arab world was present there at the beginnings of many of these large programs throughout the world. So genomic variation identified through national programs have contributed to the identification of underlying causes and risks for rare and common diseases, respectively. With rare diseases, there's been a discovery of well over 5,000 genes responsible for Mendelian diseases. And in many of these instances, these genes contribute to multiple phenotypes. This has facilitated genetic diagnosis for many patients. It provides opportunities for prenatal testing and counseling, for premarital screening, the development of new drug treatments. Cystic fibrosis would be a good example, and gene therapies in terms of diseases such as spinal muscular atrophy, for instance. And I think that when you look at rare diseases, there is a very good example of the need for each population to run genome programs, for national genome programs. And as far back as 1995, as far back as 1995, this sequencing which we did of cystic fibrosis patients in Saudi Arabia clearly identified the need for each population to enter into genome programs and discovery. At that time, patients who were diagnosed with cystic fibrosis were being sent abroad for their molecular testing, and a very large proportion of those patients had negative results. By sequencing this gene in the Saudi population, at that time we identified 13 mutations that covered about 83% of the cases that had been studied. But very importantly, there were novel mutations in the population, and at that time Delta 508 was represented about 70% of cystic fibrosis in the Caucasian population, and was actually not the most common variant in the Arab population. So a very early indication of the differences present in the population. And this was replicated when one looked at metabolic diseases as an example. Again, a small number of founder mutations could explain a large proportion of the disease cases within this particular ethnic group. So that really spurred the initiative for the Saudi Human Genome Program, and which commenced in 2014, 2015. And unlike many of the global programs, the focus was on studying rare diseases initially. And that decision was taken because this represented the low-hanging fruit and the opportunity to make the biggest impact. And on the left panel here, you can't see it clearly, but it's just a small smattering of publications of relatively large numbers of individuals that were tested for rare diseases through the Saudi Human Genome Program. And as a consequence, there are now a database of 4,183 confirmed pathogenic mutations. Many founder mutations were and unique to the Saudi population. There were many more likely pathogenic variants and variants of uncertain significance. Equally, and very importantly, many rare polymorphisms were identified through these studies, which required reclassification of pathogenic variants from other databases as benign. And there are many instances where variants in HEMD, for instance, that were considered to be pathogenic based on their frequency within the Saudi population, required to be reclassified as benign. In addition, these studies were invaluable data for the diagnosis and prevention of rare diseases in the Arab world, but also sort of provided opportunities for picking rare variants that could be utilized for looking at more common diseases. How important has that been with respect to the Saudi population? I think this is best reflected with some of the rare disease statistics which have been published by the World Economic Forum in 2020. Whilst we call these things rare diseases, in essence, collectively, they are not so rare. And the World Economic Forum stated that rare diseases affect an estimated 10% of the global population. I think this includes not just the index cases, but carriers and other members of their families. There are more than 7,000 rare diseases identified to date. And as we have the acceleration of the NGS, and as discussed through our moderator, Faisal, whilst we've identified many diseases, there are still thousands of genes and more discoveries to be made. Rare diseases affect an estimated 475 million people globally. Importantly, 80% of rare diseases are caused by genetic or genomic variants. 30% of children with the rare disease die before they reach their 5th birthday. And 1 in 3 hospital beds in pediatric hospitals are occupied by children with a rare disease. Importantly, to date, and you hope that this will be a statistic that changes because of the information coming from genome programs and from the technologies that are available, 7 years is the average time to diagnosis for many of these patients. And during that time, they see an average of 8 physicians, 4 of these primary care and 4 specialists, placing a tremendous burden on the healthcare systems. They also, in the journey to diagnosis, receive in the order of 2 to 3 misdiagnoses. And here is an opportunity for the benchmark, or molecular genetics or genomics, being the benchmark for diagnosis and for receiving a correct diagnosis. There is only approximately 5% of patients with a rare disease have a treatment available to them today. And again, one would hope that understanding the variants associated with rare diseases and studying the genes underlying these will change that particular statistic. The lifetime healthcare cost for a child with a rare disease can be conservatively in the order of $1.5 billion, but up to $10 million US dollars, depending upon the disease. When you look at the incidence of children being born with rare diseases, one can quickly see huge impact this has on the healthcare budgets of many economies. So where exactly is this burden of rare disease with respect to Saudi Arabia? And on the left-hand side here are statistics up to 2019 in terms of births within Saudi Arabia, and shows there being in the order of 500,000 to 600,000 births annually. So rare diseases affect 3.5 to 5.9% of the world's population, or equivalent to more than 300 million people. With the rare disease incidence of 4%, taking something in the middle here, and 80% of these being caused by genetic or genomic variants, one would expect that there are 19 to 20,000 babies born each year with a rare genetic disease in Saudi Arabia. As I described in my previous slide, the precise diagnosis of a rare disease takes too long, and published in the European Journal of Human Genetics was the UK experience, where over a 10-year period, looking at a little over 250,000 people, the cost involved with each of those individuals is in the order of $16,000 or $17,000, and the equivalent of $4.4 billion US dollars, or 3.4 billion pounds, for diagnosis of rare diseases. So the knowledge that we get from our genome programs and the development of technologies that can make diagnosis more efficient, once again provides opportunities for utilizing healthcare budgets more efficiently, and really represents the basis of what we would like to call precision medicine. Precision medicine is not just about diagnosis, it's also about prevention. And one of the earliest and best examples with respect to how genetics and genomics aided prevention is the example of thalassemia in Cyprus. About 15% of the Turkish Cypriot population are thalassemia carriers. 30% of spouses of individuals were found to be thalassemia carriers, with 2% of married couples both being carriers. So 2% of the marriages resulted in the birth of thalassemic children. In 1979, a high-risk family started to be screened for thalassemia, not using genetic methods, but using things like hemoglobin electrophoresis. In 1980, premarital screening was made compulsory by law for thalassemia in Cyprus, and in 1984, prenatal diagnosis was started. It wasn't until 1991 that DNA techniques were utilized rather than fetal blood sampling and hemoglobin electrophoresis. And the affected birth rates at that time showed a sharp decrease, and prenatal diagnosis started in 1984. Very importantly, as a consequence of all of this, between 1991 and 2001, there were only 5 thalassemic babies born in Cyprus, representing 1 in every 2 to 3 years. And between 2001 and 2006, 0 thalassemic babies were born. And really, this is an excellent example of how knowledge and genetics and a preventive program cannot remove the mutation from the population, but can certainly eliminate the disease from the population. So looking at that example and trying to leverage all of the information that came from the Saudi Human Genome Program and diagnostic efforts and other research work that was done in Saudi Arabia with respect to rare diseases, the incentive was there to try and utilize this information to prevent rare disease in this population. And the population of Saudi Arabia is in the order of 25 to 30 million. There are 150,000 marriages each year. As you saw earlier, 600,000 births each year. So being able to develop tools and implement programs for prevention of disease promise huge returns. Through the Saudi Human Genome Program, more than 40,000 samples from patients and families with rare inherited diseases have been screened. These represent many individuals who were screened with gene panels based upon their phenotypes. The negative individuals from those gene panels were then exome sequenced, and the collective information was used to build a database of normal and pathogenic variants within the Saudi population. As we sort of said earlier, there are 4,183 confirmed pathogenic variants, a large number of likely pathogenic variants, and these represent more than 800 inherited diseases prevalent within the Saudi population. This allowed us a couple of years ago to develop a proprietary microarray containing 7,500 disease alleles, almost half of which represent confirmed pathogenic mutations or known Saudi mutations. And this really established a foundation for premarital screening, the targeting and prevention of a large cohort of inherited diseases within Saudi Arabia. It's not sufficient to simply have these tools. It still takes a lot of commitment and the collaboration of groups of individuals within the country to be able to roll out efforts to have the impact that is possible with this knowledge. In addition to being used for carrier screening, premarital screening, and prevention programs, in utilizing the array, one could also sort of see that this information and these tools could be used to facilitate screening and early diagnosis of rare diseases. For over a decade now, some of this information that has come initially through research in specific diseases and in the latter part through programs such as the Saudi Human Genome Program, it has facilitated prenatal testing. And you can see just on the basis of the experience of one institution, that being King Faisal Specialist Hospital and Research Center, that we grew from small numbers. And in fact, I can tell you that even prior to 2011, with prenatal testing that was undertaken by our colleague, Dr. Faikar Ahmed and her group, that there were even 10 or 20 patients per year done as early as 2006, 7, 8. And this slowly grew to there being 800 cases in 2019. And our current year, I think we're somewhere between 1,250 to 1,500 cases. And as you expect, about 1/4 of the cases referred for testing are tested positive, and families are given the opportunity to make decisions with respect to continuing or terminating their pregnancy. The impact in Saudi Arabia, many families are seeking for prenatal testing in the very rare diseases where a panel of physicians and members of the community recognize that there is no treatment possible. Just a smattering of some of the alleles that were identified through our program shows you that the array can very readily pick up carriers, but in some instances, one can also sort of pick up homozygous affected individuals. So the tools that are developed through the Saudi Human Genome Program with respect to rare disease alleles do really provide opportunities for premarital screening, but also for newborn screening and early detection of disease. So precision medicine is not just about rare diseases, there's also sort of common diseases. And how do we leverage what we have done so far with respect to common diseases? A typical individual's genome varies from the reference at 4 to 5 million sites, or a little over 1 in 1,000 bases. For instance, with the 1,000 genome program, 2,504 individuals led to the discovery of 84.7 million SNPs. As of 2017, DB SNP lists over 324 million variants found in sequenced human genomes. The SNP variants have, for a considerable period of time, been used in genome-wide association studies and have been used to identify greater than 100,000 robust associations between genomic regions and common diseases. Very importantly, and I'm just citing a couple of examples to highlight a point, and that is the first African ancestry-specific APOL1 variants that protect from sleeping sickness increase the risk of kidney failure. And these variants account for 70% of the cases of non-diabetic kidney failure of African Americans. Similarly, with SLC16A11, the risk haplotype explains 20% of the prevalence in Mexican Americans. And the point that I wanted to make here is, again, like we see with rare diseases, there are variants associated with common diseases that are concentrated within populations or people of specific ethnicity. Again, highlighting the point that each community, each population, each ethnicity needs to undertake investigations to utilize the power of the genome to identify risk and prevent disease in their own populations. There are, of course, strong associations defined for a large number of polygenic diseases, Alzheimer's disease, inflammatory bowel disease, and coronary artery disease being amongst them. What has become clear also is that when you're looking at common diseases, the rare variants that you identify within populations are associated with small risks, but the cumulative risks underlie disease. And this is the incentive for the development of polygenic risk scores. And as identifying rare variants have been important for Mendelian disease, the utilization of rare variants from population to associate them with common disease and develop polygenic risk scores represents the future in terms of identifying risk and preventing disease in common disorders such as type 2 diabetes, for instance. So this is where the large-scale biobanks have become important, where the meeting of huge amounts of genomic data together with phenotype information is critical. Good examples of that being the UK Biobank and the All of Us study of the US. The All of Us study was previously the Precision Medicine Initiative from the United States. These studies have generated population-based information on variation in traits, including diseases, and facilitated the development of polygenic risk scores for prediction and prevention. What we did with data that came from the initiative to study rare diseases in Saudi Arabia, with over 40,000 individuals being studied in the database, a good proportion of those being for panels of genes associated with particular diseases, but also a large number of whole exome sequences. There was a wealth of variants present within the population that were not associated with the primary reason for which the samples were sequenced. One could nevertheless mine this information to identify the variants in the population that could then be utilized to add more power for discovery in common diseases and to develop tools for polygenic risk scores. And with the data from Saudi Arabia, what we did was to mirror what happened with the UK Biobank and to create a Saudi Biobank array. Within the UK Biobank array, there were many common variants that could be used for GWAS studies. And the power of these variants could be added to by including rare variants from the Saudi population. So by removing some alleles, some markers from the UK Biobank array and replacing them with population-specific markers, including alleles associated with rare diseases, but also rare population variants that could be used for GWAS studies. We've created a tool which I think is the future of the program in Saudi Arabia. To summarize the Saudi biobank array, relative to the UK biobank array, there are some 665,000 markers that are in common, 160,000 markers unique to the UK biobank array, but markers that were not seen within the Saudi population. A good proportion of those, of course, would be markers that are not exonic, because all of the data that we have is in fact from exomes or from panels where only the exons were sequenced. 83,000 coding region markers were unique to the Saudi population and included within the Saudi biobank array. So whilst our focus had been on rare diseases, the enormous amount of genomic information gathered still provided opportunities for mining population genomic data. And I'm going to use two examples where this has been done recently. The first is with respect to amyloidosis, or familial transthyretin amyloidosis, which is caused by mutations in the TTR gene. Amyloidosis results in a late onset of diseases associated with the accumulation of amyloid in the various organ systems. Predominantly, one sees hypertrophic cardiomyopathy as a consequence of amyloid accumulation within the myocytes of the heart, within the myocardium. So we were able to look at data from greater than 10,000 Saudi exomes from unrelated individuals blindly, and 3 TTR variants known to be associated with ATTR amyloidosis were shown to be present within the population. We also identified 3 novel TTR mutations. This is important with respect to precision medicine and shows you the value of the database in that early diagnosis and intervention facilitated by familial genetic testing of patients with ATTR amyloidosis can benefit the management of the disease. Pfizer has a drug that can be utilized to delay the onset of amyloidosis, which already, if is only present in individuals in their late 40s and 50s, with treatment, if this could be delayed by several decades, it has important significance. Similarly, looking at the coding region variants, we were able to show that pharmacogenetic variants of relevance, that only 0.8% of all of the individuals studied had no pharmacogenetic variants of relevance to this point. When you look at 1, 2, and 3, you can see the percentages and shows that the majority of the population carry 2 or 3 variants that are predicted to have an impact in terms of their pharmacogenetics. Now, extremely important with respect to using these variants in terms of polygenic risk scores, in terms of pharmacogenetics, or addressing diseases such as TTR amyloidosis, is that these studies require clinical validation. And so what we have at the moment are tools that will allow one to undertake the studies. But until the biobanks are developed and the clinical trials are done, the value of these are still limited, unlike the variants that one can associate with Mendelian diseases very, very readily. So I'd like to now sort of summarize the utilization of variants in our program with respect to precision medicine. And firstly, qualify this by saying that tumor profiling and application of targeted therapies are not discussed here at all. Of course, the information that we've gained from these variants do help these studies, but the development of somatic variant databases is separate, and I believe that the use of genomics in oncology will be the subject of other sessions within this conference. Now, the national population level genomic characterization of rare diseases substantially facilitates better diagnosis, early detection, and prevention of rare diseases. I think that case is very clear. I think it is already making a big impact with respect to diagnosis. With early detection and prevention of rare diseases, the challenge is there for the healthcare community to utilize the information and the tools to do something very valuable for the community. Programs to leverage this knowledge and capacity still need to be established and supported in Saudi Arabia. The data mining of the variants within the Saudi population do provide tools for identifying risk in common diseases and hopefully soon for the application of pharmacogenetics. With that, I'd like to acknowledge the King Abdulaziz City of Science and Technology for their support of the Saudi Human Genome Program, the King Faisal Specialist Hospital and Research Center for its support for genetics research in general over the last 2 decades, and the many, many participating physicians and researchers of the Saudi Human Genome Program and affiliated institutions in Saudi Arabia. And may I once again take this opportunity to particularly highlight the efforts of our colleague, Dr. Faisal Al-Quraya, who, through his work in Mendelian diseases and the cataloging of genes and variants associated with these disorders, is not only making a big impact for the population of Saudi Arabia and for the Gulf region, but of course, internationally has contributed so much. Thank you so much for your attention.